Welcome back, everyone, to the Deep Dive. Today, we are diving into the world of microbial metabolism.
That's right. We're going to be looking at the core principles of how these tiny organisms obtain and utilize energy.
And how they make all the stuff they need to, actually live and grow. Plus, we'll touch on just how diverse their metabolisms can be.
Which is mind-blowing. From the bottom of the ocean to volcanic vents, microbes have figured out how to survive in some really extreme places.
Talk about adaptable.
Yeah.
Okay, so before we get too deep into this, let's make sure we are all on the same page. What exactly is microbial metabolism?
Well, it's essentially all the chemical reactions that happen within a microbe to keep it alive. It has two main parts.
I'm guessing one is breaking things down and the other is building things up.
You got it. Catabolism breaks down complex molecules for energy. Think of it like generating fuel.
Okay, makes sense.
And anabolism.
That's the construction crew. It uses the energy from catabolism and basic building blocks to build all the complex molecules a cell needs.
Like proteins, DNA, all that good stuff. Exactly. It's a constant cycle of breaking down and building up.
Fueled by whatever the environment provides.
Yeah.
So you mentioned how diverse these metabolisms can be. Can you give us an idea of just how wild it gets out there?
Oh, it's incredible. We're talking about microbes using energy sources, electron donors, and carbon sources that most life can't even touch.
It's like they've figured out a way to eat anything.
Pretty much. This flexibility lets them live almost anywhere. And their metabolic activities have literally shaped our planet over billions of years.
Wow, that's powerful. But to do all this, what do microbes absolutely need? Like the non-negotiables for microbial life.
Well, first, they need liquid water. Everything happens in water. It's the solvent for all the biochemical reactions.
Makes sense. What else?
They need a source of energy that powers all those reactions.
Right. Can't do much without energy.
And they need a source of electrons. These drive many metabolic processes, especially when it comes to transferring energy.
Okay, and finally.
Nutrients. Think carbon, nitrogen, phosphorus, sulfur, and others. They are the raw materials for building everything the cell needs.
So it's like water, fuel, batteries, and building materials.
A great analogy. And these are the things scientists are looking for when they search for life on other planets.
Like on Enceladus, that moon of Saturn with the geysers.
Exactly. The fact that those geysers have liquid water and it probably interacts with rocks on the moon's core, that's a big deal.
Because it suggests they might have those energy sources, electron donors, and nutrients we just talked about.
Right. It makes Enceladus a prime target in the search for extraterrestrial life. And that life, if it exists, is likely to be microbial.
Exciting. So let's talk energy. How do microbes actually go about getting the energy they need to fuel their metabolism? This is where bioenergetics comes in, right?
Yes. Bioenergetics is all about how energy transforms and is used within cells. And a key concept is the flow of electrons.
Like electricity.
You could say that. Cells capture energy from their surroundings and need to convert it into a form they can use.
Got it. So what are the rules for this energy conversion?
The first law of thermodynamics tells us energy can't be created or destroyed, only changed from one form to another.
Okay, so like light energy can be transformed into chemical energy.
Exactly. And microbes have become masters of this kind of energy transformation.
And the molecule that's essentially the universal energy currency for all this is ATP, the adenosine triphosphate, right?
You know it. ATP is like the cell's cash. It stores energy in its phosphate bonds. When these bonds break, that energy is released to power other processes in the cell.
Like the energy requiring reactions needed for building those complex molecules.
Precisely. Those are called endergonic reactions. They need an input of energy to get going.
So ATP provides that energy.
Exactly. ATP is released from exergonic reactions, those that release energy, like breaking down glucose.
That's like burning fuel for energy, right?
A perfect example. A prime example of a super exergonic reaction is the aerobic respiration of glucose. That releases a massive amount of energy.
So all that energy is packed into a tiny sugar molecule. Amazing. Now, what about this concept of reducing power? It sounds kind of like a superpower.
Well, in a way it is. Reducing power is the cell's ability to donate electrons in these important reactions called redox reactions.
Redox. Oxidation and reduction.
That's it. And these reactions are crucial for generating energy and also for making those complex molecules we keep talking about.
So it's not just about breaking down fuel, it's also about having the right tools for building things.
Exactly. And molecules like NADH and NADPH act like tiny delivery trucks, moving electrons from one reaction to another.
Okay, I think I'm starting to get the big picture here. But how do we keep track of all these different microbes and their metabolic strategies?
Well, there are different ways to classify them based on how they get their energy and their carbon.
Like sorting them into categories?
Exactly. Based on their energy source, we have phototrophs, the light eaters. They get energy directly from sunlight.
Like plants.
Yes. Then there are chemotrophs, which get their energy from chemical reactions.
And they break down further into...
Chemorganotrophs and chemolithotrophs. Chemorganotrophs break down organic chemicals like sugars and amino acids.
Okay, so they get their energy from things we would consider food. What about the chemolithotrophs?
They get energy from inorganic chemicals. Things like hydrogen gas, hydrogen sulfide, ferrous iron, ammonia.
Wow, so they're essentially eating rocks and gases. That's mind-blowing.
It is. They've tapped into energy sources that are completely unavailable to us.
And how do we categorize microbes based on their carbon source?
We have heterotrophs, which get their carbon from organic compounds. Like chemorganotrophs, they're eating what we would think of as food.
Got it. So what about the autotrophs?
They use carbon dioxide as their carbon source. They can fix carbon dioxide and turn it into organic molecules.
Like plants again?
Exactly. Most chemolithotrophs and phototrophs are autotrophs. They're the primary producers, the base of many food webs.
Okay, so we've talked about energy and carbon sources. You also mentioned that electron transfer reactions are crucial for all this. Could you explain why?
Redox reactions are at the heart of how cells get energy. One molecule loses electrons, another gains those electrons. This movement releases energy that the cell can use.
So it's like a cascade of energy being passed down.
In a way, yes. And each molecule has a different tendency to gain or lose those electrons.
So some are better at grabbing those electrons than others.
Right. And that's measured by their reduction potential. The bigger the difference in reduction potential between two molecules, the more energy is released when electrons move between them.
And this energy release is directly related to the free energy available to the cell, right?
Absolutely. There's an equation that links the change in reduction potential to the change in free energy. It tells us how much energy a particular electron transfer can provide.
That's pretty neat. So cells capture this released energy by forming energy-rich compounds. We talked about ATP, but what are some other examples?
You have things like phosphoenolpyruvate, 1-chala-3 bisphosphoglycerate, acetyl phosphate, acetyl-CoA.
Those are quite a mouthful.
They all have high energy bonds that, when broken, release energy for the cell to use. But ATP is the most important and versatile.
It's like the main currency of the cell.
Exactly. And there are three main ways ATP is made: substrate-level phosphorylation, oxidative phosphorylation, and photophosphorylation.
Could you break those down for us?
Sure. Substrate-level phosphorylation is a direct transfer of a phosphate group from a molecule to ADP, making ATP.
Okay. So it's like a direct energy transfer.
Right. Oxidative phosphorylation uses an electron transport chain to generate a proton gradient across a membrane.
We talked about that a bit earlier, right?
Yes, and that proton gradient is used by ATP synthase to make ATP.
Like water flowing downhill and turning a turbine.
Exactly. Finally, photophosphorylation is used by phototrophs to generate ATP using light energy.
So it's essentially photosynthesis making ATP.
Exactly. Now, we can't forget the amazing enzymes that make all these reactions happen.
They're the workhorses of the cell.
Exactly. Enzymes are biological catalysts. They speed up biochemical reactions.
So they make things happen much faster than they would otherwise.
Exactly. And they do this by lowering the activation energy that's the energy needed to kickstart a reaction.
So they make it easier for reactions to occur.
Precisely. And each enzyme is super specific for its target molecule. It's like a lock and key.
That makes sense. And what about coenzymes and prosthetic groups? How do they fit in?
Many enzymes need helpers to function correctly. These helpers can be metal ions or organic molecules.
Like sidekicks.
You'd think of it that way. If the organic molecule is loosely bound to the enzyme, it's called a coenzyme.
Okay. And an example is?
NAD plus NADH and NADP plus NADPH are good examples. They act like electron shuttles carrying electrons between reactions.
Got it. And what about the tightly bound organic molecules?
Those are called prosthetic groups. They're permanently attached to the enzyme and are essential for its function.
Like a built-in tool. Okay, so now we've got a pretty good grasp of the basics. Let's dive deeper into how specific microbes get their energy. First up, the chemorganotrophs. How do they utilize those organic molecules?
Chemorganotrophs get both their energy and carbon from organic molecules. They use a near-universal pathway called glycolysis to break down glucose.
That's a familiar name from biology class. What does glycolysis do?
It breaks down one glucose molecule into two pyruvate molecules.
Okay, so A 6-carbon sugar is split into 2 3-carbon molecules.
Exactly. And this process happens in 2 stages.
What are the stages?
Stage 1 requires an investment of energy. 2 ATP molecules are used.
So the cell has to spend some energy to get the process going.
Right. Stage 2 is the payoff phase. 4 ATP molecules are produced along with 2 NADH molecules.
So a net gain of 2 ATP and 2 NADH, not bad. But you mentioned earlier that glycolysis on its own isn't balanced in terms of redox reactions. Can you explain that?
Sure. Glycolysis produces NADH, but it doesn't have a way to regenerate NAD plus using an external electron acceptor.
So it needs another process to keep things going.
Exactly. Either respiration, if an electron acceptor is available, or fermentation, if not.
Got it. So what happens to those pyruvate molecules produced by glycolysis?
If the organism can respire, they head to the citric acid cycle, also known as the Krebs cycle.
Ah, another familiar name. Refresh my memory on what the citric acid cycle does.
It completes the oxidation of those carbon atoms from glucose all the way to carbon dioxide.
So it's like the final breakdown stage.
Precisely. And it generates a lot of energy carriers. 1 ATP, 3 NADH, and 1 FADH2 per cycle.
You get 2 cycles per glucose molecule, right?
That's right. So if we add it all up from glycolysis and the citric acid cycle, we get a total of 4 ATP, 10 NADH, and 2 FADH2.
That's a lot of energy. But the citric acid cycle isn't just about energy production, right?
You're right. It's also crucial for biosynthesis. Many intermediates in the cycle are starting materials for building other molecules.
Like what?
Amino acids for proteins, cytochromes for the electron transport chain, chlorophyll for photosynthesis, even fatty acids.
So it's like a central hub for both breaking down and building up molecules.
Exactly. And there's a modified version of the citric acid cycle called the glyoxylate cycle.
What does that do?
It allows organisms to grow on 2-carbon compounds like acetate. It bypasses some steps of the citric acid cycle, allowing the cell to conserve carbon and build essential molecules.
Interesting. Okay, so that's what happens when oxygen is present. But what about when there's no oxygen? That's where fermentation comes in, right?
Exactly. Fermentation is anaerobic. It doesn't need oxygen or any external electron acceptor.
So how does it work?
Organic compounds act as both the electron donor and the acceptor. And all the ATP comes from substrate-level phosphorylation. No electron transport chain involved.
So how is the NADH produced during glycolysis recycled back to NAD plus without an external electron acceptor?
Pyruvate, or a molecule derived from it, accepts those electrons from NADH, regenerating NAD plus for glycolysis to continue.
Ah, so pyruvate acts as the electron sink.
Precisely. And this reduction of pyruvate leads to those fermentation products we all know and love.
Like lactic acid in yogurt or ethanol in beer.
Exactly. Lactic acid bacteria produce lactic acid, while yeast produce ethanol and carbon dioxide.
So different microbes produce different fermentation products.
Right. And these fermentation pathways allow microbes to survive in all sorts of oxygen-free environments.
Pretty clever. Okay, let's move beyond just consuming organic molecules. What about electron transport and the diversity of metabolisms out there?
Well, we've touched on respiration, where electrons move from reduced electron donors to external electron acceptors.
And anaerobic respiration, oxygen is that final electron acceptor.
Exactly. And glycolysis and the citric acid cycle produce those electron carriers, NADH and FADH2.
Right, and those donate their electrons to the electron transport chain.
Precisely. The ETC is a series of protein complexes embedded in a membrane.
Where is this membrane located?
It's in the cytoplasmic membrane of prokaryotes and the inner mitochondrial membrane of eukaryotes.
And what happens as electrons move down this chain?
Energy is released and used to pump protons across the membrane, creating an electrochemical gradient.
And that gradient is the proton motive force, right?
You got it. And that proton motive force drives ATP synthesis through oxidative phosphorylation.
So it's all connected.
Absolutely. The potential energy stored in that gradient is harnessed by ATP synthase to make ATP.
It's like a microscopic power plant. What are the key players in this electron transport chain?
There are several types of molecules, all arranged in order of increasing affinity for electrons. You have NADH dehydrogenases, flavoproteins, iron-sulfur proteins, cytochromes, and ketones.
Those names sound vaguely familiar from biochemistry.
They all work together to move those electrons down the chain and pump those protons across the membrane.
You mentioned Paracoccus denitrificans as an example. What's special about its electron transport chain?
It's very similar to the one found in the mitochondria of eukaryotes, making it a valuable model for studying respiration.
And what about ATP synthase? How does it actually use that proton flow to make ATP?
ATP synthase is an amazing molecular machine. It has two main parts: the F0 subunit embedded in the membrane and the F1 subunit sticking out into the cytoplasm or mitochondrial matrix.
And the F0 subunit forms a channel for protons, right?
Right. As protons flow through, they make the F0 subunit rotate. This rotation turns the F1 subunit, which then catalyzes the formation of ATP.
So it's a tiny rotary engine?
A perfect analogy. Now, we've talked about aerobic respiration. What happens when there's no oxygen?
That's when anaerobic respiration takes over, right?
You got it. It follows the same principles, but instead of oxygen, a different molecule acts as the final electron acceptor.
Like what?
Nitrate, sulfate, elemental sulfur, even some organic compounds.
So there's quite a bit of flexibility depending on the environment.
Absolutely. The energy yield depends on the difference in reduction potential between the electron donor and the final acceptor. Oxygen is the most efficient, but microbes have found ways to use other molecules when oxygen isn't around.
It's all about adapting to survive. Now let's talk about those amazing microbes that eat rocks, the chemolithotrophs. How do they get their energy?
They oxidize inorganic electron donors, things like hydrogen sulfide, hydrogen gas, ferrous iron, ammonia.
And they use that energy to power their metabolism.
Exactly. And most of them are also autotrophs, meaning they use carbon dioxide as their carbon source.
So they really are like rock-eating plants.
In a way, yes. They oxidize those inorganic compounds, feed the electrons into an electron transport chain, and use that to generate ATP.
And many of them can switch between aerobic and anaerobic respiration depending on the availability of oxygen.
That's right. They're very adaptable, and some chemolithotrophs have to use reverse electron transport.
That sounds like it's going against the flow. Why would they do that?
They need it to generate NADH, which is essential for carbon fixation. Sometimes their electron donor isn't powerful enough, so they have to use energy from the proton motive force to push those electrons uphill.
That's incredible. Okay, let's move on to the other major players in primary production, the phototrophs.
These are the organisms that convert light energy into chemical energy through photophosphorylation.
That's photosynthesis.
Exactly. And there are two types: oxygenic and unoxygenic.
Oxygenic photosynthesis is what plants and algae do, right?
Exactly. They use water as the electron donor and produce oxygen as a byproduct.
And anoxygenic photosynthesis.
That's found in certain bacteria. They use electron donors other than water and don't produce oxygen.
Okay, so it's still photosynthesis, but with a different starting point.
Right. And they both use specialized pigments like chlorophyll to capture that light energy.
And that energy is used to generate ATP, right?
Exactly. The light energy excites electrons in the pigments. These excited electrons then flow through an electron transport chain, creating a proton motive force that powers ATP synthase.
And you mentioned cyclic photophosphorylation in purple bacteria. What's different about that?
The electrons cycle back to the starting point, so it's a closed loop. As long as there's light, they keep cycling and generating ATP.
So they don't need an external electron donor for this process.
Exactly. But anoxygenic phototrophs still need an external electron donor for other metabolic processes, and they often have to use reverse electron transport to make NADH for carbon fixation.
Now let's talk about autotrophy, using carbon dioxide as the carbon source. What pathways do microbes use for this?
The most common is the Calvin cycle, used by oxygenic phototrophs, some phototrophic bacteria, most chemolithotrophic bacteria, and some archaea.
That's the one I vaguely remember from biology class. Can you remind me how it works?
It's a cycle with three main phases. First, carbon dioxide is fixed into an organic molecule by the enzyme rubisco.
Okay, so that's how inorganic carbon gets incorporated into the cycle.
Then, in the reduction phase, that molecule is reduced using ATP and NADPH, forming a molecule that can be used to make glucose.
In the third phase,
That's the regeneration phase, where the initial carbon dioxide acceptor molecule is remade so the cycle can continue.
it's like a carefully choreographed dance of molecules.
A perfect description. And it takes a lot of energy, 12 NADPH and 18 ATP, to make one glucose molecule.
Wow, that's quite an investment. Are there any other carbon fixation pathways?
Yes, some bacteria and archaea use alternative pathways, like the Wujong-Dahl pathway.
And these alternative pathways might be more efficient in certain environments.
Exactly. Okay, last but not least, we have to talk about nitrogen fixation.
That's converting nitrogen gas into a form that life can use, right?
Right. Atmospheric nitrogen is very stable and unusable by most organisms.
So how do microbes make it usable?
They use an enzyme complex called nitrogenase to convert nitrogen gas into ammonia.
And ammonia can then be used to build other molecules.
Exactly, especially amino acids. But this process takes a lot of energy and is very sensitive to oxygen.
So how do nitrogen-fixing microbes protect nitrogenase from oxygen?
They've developed various strategies. Azotobacter produces a slime layer that limits oxygen diffusion.
Anabolism.
Anabaena forms specialized cells called heterocysts that have an oxygen-free environment where nitrogenase can work safely.
So they found ways to work around the challenges.
Absolutely. Now, we've seen how microbes get energy, carbon, and nitrogen. But how do they use these to build all the stuff they need? That's where biosynthesis comes in.
So we're talking about making the building blocks of life.
Exactly. It all requires energy, typically from ATP and the raw materials from the environment or from other metabolic processes.
Let's start with polysaccharides like glycogen and peptidoglycan. How are they made?
They're synthesized from activated sugar molecules like glucose. These activated sugars are added to existing chains, making the polysaccharide longer.
So it's like building a chain link by link.
Exactly. And what if the cell needs glucose but can't get it from its surroundings?
That's where gluconeogenesis comes in.
Exactly. It's essentially glycolysis in reverse. It makes glucose from non-carbohydrate precursors.
So the cell can make its own glucose if needed.
Right. And then there's the pentose phosphate pathway.
We talked about that a bit earlier. It makes those 5 carbon sugars for nucleic acids.
Yes, but it also does much more. It produces NADPH for biosynthesis and interconverts sugars with 3 to 7 carbon atoms.
So it's a multi-talented pathway.
Mm.
What about making proteins? How are those amino acids put together?
The carbon skeletons for amino acids come from intermediates in glycolysis and the citric acid cycle. Then, an amino group is added, usually from glutamate or glutamine, through transamination.
So it's like taking a pre-made carbon skeleton and adding the amino group.
Exactly. And the amino group ultimately comes from ammonia, which is incorporated by enzymes like glutamate dehydrogenase and glutamine synthetase.
So it's all interconnected. And what about nucleotides, the building blocks of DNA and RNA?
Nucleotide biosynthesis is complex. You have purines like adenine and guanine, and pyrimidines like cytosine, thymine, and uracil. They're synthesized through different pathways using various precursors.
It sounds very intricate.
It is. And ribonucleotides, those used in RNA, are made first. Then, some are converted into deoxyribonucleotides for DNA.
Okay, so it's RNA first, then DNA. Makes sense. Finally, let's talk about lipids.
Lipids are made by adding fatty acids to a glycerol backbone.
And how are those fatty acids made?
In bacteria and eukaryotes, they're made by adding 2 carbon units to a growing chain.
And this results in those even-numbered fatty acids we hear about.
Right. But modifications can happen, creating unsaturated, branched, or odd-numbered fatty acids.
So there's some variation depending on the organism.
Exactly. And then you have the archaea, which use isoprenoids instead of fatty acids in their lipids.
So their membrane lipids are quite different.
They are indeed.
Wow, that was a whirlwind tour of microbial metabolism. We covered so much ground, from the basics of energy and building blocks to the intricacies of various metabolic pathways.
It's amazing to think that all this complexity is happening inside these tiny organisms.
I know. And it really highlights the central role microbes play in the world. They're the ultimate recyclers, transformers, and producers.
They drive so many processes that are essential for life as we know it, and the fact that they can thrive in such diverse environments is truly remarkable.
It makes you wonder what other metabolic secrets they're hiding, especially in those extreme environments we haven't explored much yet. What do you think might be the most unexpected metabolic process out there waiting to be discovered?
That's a fantastic question. The possibilities are endless. Maybe there are microbes that can use energy sources we haven't even thought of yet. Or maybe they've developed completely novel ways to fix carbon or nitrogen.
It's exciting to think about all the mysteries that are still out there.
Absolutely. The study of microbial metabolism is full of surprises.
Thank you so much for taking us on this incredible journey into the world of microbial metabolism. I've learned so much.
It was my pleasure. It's a topic that I'm always excited to talk about.
And that brings us to the end of this deep dive into microbial metabolism. We've covered all the major points, theories, findings, and examples from the provided chapter.
From energy acquisition to biosynthesis, we've explored the amazing diversity and adaptability of microbial metabolism.
And it's clear that these tiny organisms play a huge role in shaping our world.
Absolutely. Their metabolic capabilities are essential for life as we know it.
Thanks for joining us, everyone, and be sure to tune in next time for another deep dive into the fascinating world of science.